My name is Mr. Sexually, and today I'm going to go over the personal protective equipment that you're going to need, some dissection techniques, and the steps for your brain dissection that you guys are going to be starting. So the first thing we're going to talk about is PPE, which stands for Personal Protective Equipment. Purpose of the personal protective equipment is to make sure that it does not allow for the spread of harmful or disease-causing agents from one person to another. Now, you guys aren't going to be working with dead or sick people, but you are going to be working with preserved brain specimens. These brains are preserved with certain types of chemicals that can cause you to be sick. So we put on these particular pieces of equipment in order to prevent you from getting sick. The first thing that you're going to put on is called a lab apron. That also prevents your clothes from getting dirty as you are doing the dissection. The next thing that you're going to go ahead and put on or don is called the gloves. So you place one glove on each hand, like so. Now, if you are wearing long sleeves, I highly recommend that you take those sleeves and you want to roll them up, so therefore nothing gets into the brain juice as we are dissecting. The last thing that you're going to want to put on is going to be eye protection, like so. Now, if you have hair that is down to your shoulders or longer, you're going to want to make sure that it is tied back with a hair tie or a ponytail to ensure that when you are bending over looking at your specimen that it does not get into your brain. To get started with your dissection, there's a few things that you're going to need. You're going to need a dissecting tray that you're going to put your specimen into. You are going to need a scalpel. Make sure you're very careful handling this because it is sharp. And the last thing you're going to use is called a dissecting probe. Now, when you are using these various different things, you want to make sure that only one person uses these various tools at a time, and you want to make sure that you are being very careful so therefore you do not injure yourself or injure someone else around you. It is really difficult to get hurt inside of a laboratory setting, but if you don't follow the safety rules that have been set up, that's when injuries are going to happen. I'm going to demonstrate now how to go ahead and properly hold the scalpel. Now, there is a right and wrong way to hold your scalpel. With this particular device, the rounded edge here is going to be the sharp side or the blade, and what we have up here, which is the flatter or smoother side, is going to be the dull end. Now, you do not want to hold your scalpel like you're going to be sawing or chopping into something. You want to gently rest the scalpel in your hand, taking your pointer or your index finger and placing it over the smooth or flat end of the blade. This is going to give you a lot of control with your scalpel as you are making your incisions into your specimen. So I'm going to demonstrate how to make an incision on this apple. So you want to go ahead and take the apple. Now, when you are holding the apple, holding the specimen, and the apple's going to be our specimen, you want to place it so therefore you are never cutting towards your body, because that's how injuries can happen. You always want to be making the incision and making the cut away from your body. So as you can see, I've taken it, moving away from my body itself. I take the scalpel and I'm going to make the incision while I cut away from my body and also cutting away from anyone in my lab group. Now, as you can see, I made one clear cut, one clean incision that went deep into my specimen. So as you take your scalpel across your specimen, you just want to kind of let it rest and let your blade do the work for you. So cut from one end to the other, always cutting away from your body. Make sure that your lab partner or your group is not standing in the direction that you are cutting your specimen. Now, if you see where your first incision is going to be, we want to make this incision larger. The way that we're going to do this is let your blade rest within that cut and gently push down deeper into the specimen to further separate it. So always make sure you hold onto that flat end. Now this is going to be until you can get all the way through. And this is the exact same type of cutting action that we are going to do with the brain. So as you get the specimen more and more cut, go ahead and try and loosen it so you can see inside, so you know where to make your next incision. Now if you look at the edges of the apple, you can see how clean and nicely cut they are. There are no jagged edges. This is going to allow you to preserve the inside of the specimen so we can see it in greater detail. All right, so we're going to get started with our brain dissection. The first thing that you're going to do is you're going to take your brain out of the dissection jar, and you want to place it into your tray. Now, there's going to be a lot of juice left over in your dissection jar, so if you want to take a little paper towel and just kind of lightly dry off your brain, so therefore you're able to see what you're working with. Now, make sure you have your PP on during this whole entire time to prevent your hands becoming contaminated with chemicals. So once you look at the sheep brain, what you notice is that you'll see these individual different folds. Those folds are designed to go ahead and add extra area, extra nervous tissue. Now the brain is very special. It's the only organ inside of our entire body that's going to process information. It's going to then send a signal back telling our body how to respond. So each of these different parts of the brain represent a particular function that's going to be carried out in that particular region. So your objective is going to be identifying those various different regions of the brain, different parts of the brain, and then we're going to see what function they are going to have within our body. So the first thing that we have to be able to identify is going to be the brain stem. So using your pre-made flags, what you're going to do is just take your flag and put it into that particular region of the brain. So here you can see I have the brainstem. The brainstem is going to be located on the bottom part of the brain. It looks like a little tiny cord that's going to come off the brain itself. So you just want to take your flag and place it directly into that particular region. The next structure that we want to be able to identify is called the cerebellum. Now the cerebellum, another name for the cerebellum is going to be the tiny brain. So cerebellum is going to be the region that's not connected to the main part of the brain, but it's going to be anterior or it's going to be posterior and dorsal, so to the back. So we just place the cerebellum flag in like so. Now the next and last region of the brain that we're going to have, so over the three main regions, is called the cerebrum. The cerebrum is going to include all of the major lobes that we're going to look at in more detail. So the cerebrum is going to be this main portion that we see at the very top. So those are the three main regions that we have within the brain, three main parts. So now we're going to start on the main dissection, or the main identification of the cerebrum within our brain. So we want to take the cerebrum and we want to take where the frontal lobes are located in the front part of the brain away from the brainstem. We want to point it facing away from us. So now we're going to identify where we have what are called the frontal lobes, the parietal lobes, the temporal lobes, and the occipital lobes. Lobes is just going to be the reference for a certain rounded area of the brain itself. Now if you look at your brain, you're going to notice that there is a section, a line that's running all the way down the middle, separating the cerebrum. Now this means that the left and the right side are going to be the exact same. So therefore, when we talk about the left and right, it's going to be from the frontal lobes facing the front. So therefore, this side would be the right frontal lobe, and this would be the left frontal lobe, located in the front region of the brain. Then moving our way back, from the frontal lobes, we have what are called the parietal lobes. Those are located in this particular region on the top part of the brain. So once again, what we have the right parietal lobe located here, and then we have the left parietal lobe. Moving down away from the parietal lobes on either side, and this is what would be the part of the brain that would be controlling auditory, hearing different things, and processing that information. So therefore, on either side of your ears are called the temporal lobes. So on either side of the brain are called the temporal lobes. So the left temporal lobe located here, and the right temporal lobe located here. Now, in order to see this very last lobe that we have within our brain, we're going to have to look at it upside down. So if you look at the brain upside down, you'll notice this is another lobed region that we can see in our brain, and that is called the occipital lobe. Now, if you look very closely, when I took the brain and I flipped it upside down, what was on the left side now looks like it's going to be the right. So this is still going to be the left occipital lobe, and the right, which is what looks like the left, is actually the right occipital lobe. So here are the 4 different lobes of the brain. To look at the next part of the brain we want to study, we want to take the brain and we want to turn it upside down. So therefore, with the frontal lobe still facing away from you, you're going to take the brain and set it like so. So on the bottom portion, we're able to see certain individual structures. Up here at the region of the frontal lobes, the bottom portion, we see these kind of things that look like raised edges that are white. Those are called the olfactory bulbs. So on either side, you have 2 olfactory bulbs. Now this region right here is extremely important. It is called your optic nerve. Now the nerves itself are going to kind of split off in a Y shape, but as you can see, my optic nerve has been severed. This is where information from your eyes is going to be transmitted into your brain. So here we have the olfactory bulbs, these regions, and the optic nerve. Okay, we are now ready to go ahead and start cutting into our brain. The first thing we want to do is to go ahead and remove all of our identification flags that we have used. You can set those to the side. So now you want to go ahead and take your scalpel. Remember how to hold your scalpel. You want to hold it and resting in the palm of your hand, taking your pointer finger, putting it over the dull end of the blade, holding the specimen with your other hand, and cutting away from your body. You're going to start at one end of the brain stem and you're going to cut right down the middle. Maybe. Just like so. So once again, find where that incision was and go ahead and make it a little bit larger. So you want to cut your brain in half, like so. As you saw when I was cutting, this part of the brain came off. This was due to the dense connective tissue that was connecting it to the brain itself. But located within this connective tissue right here is actually going to be our pituitary gland. So the pituitary gland helps in maintaining hormone balances within our body. If that area gives you a little bit of difficulty as you are trying to make your incision into your brain, make sure you ask for help to get it fully removed. Now looking at the inner parts of the brain, separating the left and the right sides. Now once again, everything's going to be a little bit flipped. So this is the right side of the brain and this is going to be the left side of the brain. What we can see is that if we find where that optic nerve was located right here, and we take the brain and we separate it in two, looking on the inside, we can now see that the optic nerve is going to be located right here. On the inner part of the brain, that's what we're going to refer to as the optic chiasm. So when we're talking about a chiasm, it means it's simply going to be a narrow opening. So optic chiasm is on the inner part of the brain coming from the optic nerve. All right, we're going to go ahead and identify the last three main parts of the brain for this particular dissection. That's going to be the corpus callosum, the thalamus, and the hypothalamus. The corpus callosum is going to be the region that connects the brain, the left and right parts of the brain, and allows for communication between those two individual parts. If you look here, you can see a kind of lighter area that is outlined in the middle part of the brain, and that's what we call the corpus callosum. It's just going to be a bundle of fibers that allows for our left side of our brain to talk to the right side of our brain. We then have this particular region of the brain that is called the thalamus. So this is going to be a major regulatory part of the brain, getting information from different regions and then sending it out, telling it where to go within our body. So this is called the thalamus. Now directly below the thalamus, we have what is called the hypothalamus. This is also going to work with the pituitary gland to maintain hormone levels inside of our body. Congratulations, you have finished your brain dissection, and it's now time to start cleaning up. So to clean up, what you're going to do is remove the remainder of your flags that you have within your brain. At the beginning of your lab, you probably wanted to go ahead and put these brown paper towels in the bottom of your dissection tray, because all you now have to do is take those paper towels, wrap your brain up, and put it in the specified waste container. After you are finished, you are then going to go ahead and take a cleaning wipe or cleaning spray. You want to wipe out the complete inside of your dissecting tray to make sure that none of those preservatives or chemicals are going to linger around. After you have wiped off your tray, you want to go ahead and wipe off the space where you worked. And also wipe off your dissection tools. So your scalpel, be very careful, and your dissecting probe. Now that you have everything finished, it's time for you to go ahead and take off your PPE. Be very careful as you are taking off your gloves to make sure you do not come in contact with your skin. And make sure to go and wash your hands or use hand sanitizer.